Hi, everyone. I'm Thomas Goralski. I'm a fourth year PhD candidate here at the Andel Institute. I'm in the Henderson Neurodegenerative Disease Lab, and I'm very excited to here to talk to you today about the role of protein aggregates in Parkinson's disease. But before we get into that, I wanted to kind of introduce this really key concept because I'm a cell and molecular biologist, and that sounds like a whole lot of jargon that may not make sense to everyone. So I wanted to break down what that is so you understand what it is that I'm really asking about biology. And so let's break it down. So when I say cellular, what I'm talking about is the fundamental unit of a living being. These are the things that make up each and every one of us and all living things on Earth. We have these fundamental units called cells. And when I refer to molecular, what I'm talking about is the fundamental unit of chemistry. So molecules are to chemistry what cells are to biology. And these are specifically the smallest part of bonded atoms that can chemically react. So these are just very small objects made of bonded atoms that react to do things. And of course, biology is the study of life. And so in summary, what I am as a cell and molecular biologist is I study how molecules and cells work in living things. And so what I'm really interested in is how these protein aggregates in Parkinson's disease affect the molecules and the cells in patients with Parkinson's. And so there's this central dogma in molecular biology that's really important to understand that kind of really frames my question that I've asked and how I approached it. And so first off, we have DNA. This is deoxyribonucleic acid, which really just is probably what you've heard of, referred to as your genes, right? It's what you inherit from your parents, and it kind of determines your kind of trajectory over your lifespan of all the things your body can do. This DNA gets transcribed into what we call messenger RNA, which is just a different type of molecule that coincides to the genes in the DNA. This messenger RNA then gets translated into proteins, which are doing things in the cell. So what we have here is really a system where we understand that proteins are what's doing things in the cell. DNA is all of the things that a cell can do, and RNA is really kind of all the things that a cell is trying to do. So in response to its environment and the signals it receives, this is what the cell is trying to do in response to those. And so what I really am focused on in my PhD project is these proteins that are involved in Parkinson's disease. And that is the Lewy pathology, which is comprised of misfolded and aggregated alpha-synuclein, which is a protein that's present in many living organisms. And this protein, when it misfolds, it takes on two major forms. We call this a Lewy body, which is here and here, which are these large, dense aggregates that form in the main part of a neuron, and also in some other cell types. And additionally, these Lewy neurites, which are these long, thinner aggregates that are almost spindle-like. And what's really interesting about these proteins is that we really don't know a lot about them historically.
Did you say that the protein, when it forms these bodies, are misfolded?
Yeah, they are misfolded.
Okay.
So the proteins all have what we call a conformation, but basically they take on a shape, right, based off their sequence and other factors. And so what happens to the alpha-synuclein in this context is it's taken on the wrong shape. And what happens is when it takes on that wrong shape, other alpha-synuclein proteins come in and interact, and it aggregates further and further and builds out these spindle-like structures in these large, dense aggregates. Does that answer your question?
Yes, thanks.
Yeah, of course. And so this puts Parkinson's disease in the realm of what we call proteinopathies. And specifically, it's over here on this end of the spectrum where alpha-synuclein being a pathological protein involved in the disease, this is Parkinson's disease dementia, and dementia with Lewy bodies all have this proteinaceous aggregate that occurs in the pathology. And we actually neuropathologically diagnose Parkinson's disease based off the presence of this Lewy pathology. This misfolded alpha-synuclein is how we confirm the diagnosis of Parkinson's disease post-mortem. And additionally, it's really important to understand that many neuronal cell types have these pathological aggregates. It's not just the dopaminergic neurons, but actually throughout much of the brain, there are cells that can have this pathology. And not all patients with Parkinson's disease have this Lewy pathology, and there's many factors that contribute to that, but many patients do. And so this pathology is understood to progress through the brain. And so what kind of this graphic is meant to demonstrate is that while yes, in the substantia nigra we do observe pathology, it also happens throughout most of the rest of the brain as the disease progresses throughout the course of the disease process. And like I had just described earlier, this pathology is forming and spreading to our best understanding through this process where we have this alpha-synuclein, which for unknown reasons takes on this misfolded conformation. We still haven't figured out why this occurs, just that it does. And that this misfolded alpha-synuclein interacts with the healthy, non-pathologic alpha-synuclein and leads to this templating effect where it aggregates and builds out these large aggregates that we call loop. And so what I want to highlight here is that it really is true that not all neurons have pathology in Parkinson's disease. So on the right here is actually an image of a piece of brain tissue, and in red these are neurons, right? And you can see from this same brain, in yellow, there's the marker for this Lewy pathology, which is phosphoserine 129 of alpha-synuclein. That specific phrase doesn't really matter. Just know that the yellow is where the Lewy pathology is. You can see in the same piece of brain tissue that there are neurons very nearby each other within the brain that have pathology and do not have pathology. And so what I'm really interested in is why is that the case? So why do some neurons get these aggregates and why don't others? And additionally to that, so here you have the neurons with the Lewy pathology, which we call vulnerable, and we have neurons in the same brain without pathology, which we call resilient. And I want to understand what factors contribute to this. So why do some neurons get this pathology and why do others not? The other part of this that the field hadn't really understood very well up until my work in my PhD is exactly what alterations happen in these neurons that I'm calling here vulnerable. We didn't know, is this a protective process? Is this trying to help cells that are, sick and dying? Or is this actually contributing to the degeneration that we observe in the disease? It really wasn't clear exactly how these contributed. And so to address this, what I wanted to understand is when we have this misfolded alpha-synuclein as a protein, what effect does it have on the cells here at what the cell is trying to do? So how is the cell responding in the mRNA? And I did this with a really cool technology called spatial transcriptomics, which I'm going to talk to you about in just a little bit after I kind of introduce this concept, because I was really interested in understanding the role of this in the cortex, because like I said, it's not just dopaminergic neurons that get affected by this Lewy pathology. It happens throughout the brain in many different cell types. And in the cortex, we actually see these aggregates develop in the cortex as pathology enters the cortex and as symptoms like dementia and cognitive dysfunction occur in patients. And so what I really wanted to understand is the kind of molecular underpinning of this new symptom onset. And so there's many cells, neuronal cell types that occur, and we classify them on many different factors, one of them being what those cells do. So here we have on the left glutamatergic and on the right GABAergic. And what glutamatergic and GABAergic refer to are the neurotransmitters, those signals the cells are sending to each other back and forth. And on the left here, the glutamatergic, these neurons, when they signal, they cause the cells that they're talking to fire. They signal for them to be active. And these GABAergic neurons, they inhibit cells. So when they signal to other cells, they tend to make them less active. And so that's one way we classify neurons, but then we can also subclassify them based on how they look. And so there's as many neuronal cell types in the brain as you can probably think of. There's many thousands of them. And I wanted to understand what factors contribute to the specific vulnerabilities of specific cell types in the brain. And so by looking at the mRNA through the spatial transcriptomics approach that I brought up earlier, we're able to kind of understand how these cells are responding to that pathological protein, as well as be able to kind of identify what cell types they are. And so I'm going to talk a little bit about this really cool technology that we now have access to here at our institute, because this was really kind of the major stopgap to addressing these critical questions in the field, was access to technology exactly like this. Because understanding the difference between cells that have pathology, so these red cells that have the yellow, and capturing from the same brain as the neurons that don't have pathology, wasn't something we could do before this technology existed. So here you're seeing an actual brain. This is a misfolded alpha-synuclein on neurons. And what we're able to do is, as you can see here, I'm highlighting neurons that have pathology and don't have pathology. what we're able to do is we capture all of the RNA from these two groups of cells separately. So we capture all of the RNA from cells that have pathology, and then separately we capture all of the RNA from cells that don't have pathology. And this allows us to really interrogate those questions of what is the protein doing and what cells are affected by the pathology. And so when we capture all of this, we're able to really interrogate those questions, and we can do it for many different regions of the brain that we're interested in. And this actually resulted in our understanding of how pathology impacts the brain, specifically neurons, and exactly in the cortex, which neurons seem to be vulnerable and resilient. And this is a really important concept because when we understand resilient and vulnerable neurons, we can start to look into the factors that made them that way in the first place. Because we have an understanding of what these neurons look like in healthy tissue. And so by understanding which neurons are vulnerable and resilient to pathology, we can understand what made them that way. And so what we found was that the vulnerable neurons are these layer 5 intratelencephalic neurons. So go ahead and try and say that 5 times fast. I did that too. And the layer 6B neurons are much easier. And then resilient neurons are these inhibitory neurons, so the cells that tend to make other cells not as active, as well as these pyramidal tract neurons. And so what I'm really interested in this latter part of my PhD is really interrogating exactly what the differences are between these neurons kind of in at a baseline level, and understanding these factors that are contributing to their resilience and their vulnerability. And so then we can target those in the cells that seem to be dying. Because what we actually found is that in the cells with aggregates, what we observed is that there's evidence that there is DNA and RNA damage. So those like really key parts of the cell that gives us all the proteins that allows us all of the functions in the cell. There's extensive damage going on in the cells with these aggregates. And additionally, there's a strong immune response. So the immune system is how we get better when we're sick from like a viral pathogen, but there's also another part of the immune response which helps us maintain our body's homeostasis, and it responds to cells that are dying. Additionally, what we see is that these cells actually lose their cytoskeleton. So this is kind of like the skeleton of a cell. So if you think about your bones that like keep you upright, these are the bones that keep a cell in its shape and allow it to maintain its physiological function. They lose a lot of the key components of their cytoskeleton. Their endolysosome, which is kind of like the highway of a cell, it's how cells traffic things around. There's all sorts of damage and alteration that occurs there. The synapses, they lose their synapses. So the synapses are the connections between neurons, and it's how they communicate, which is essential to their function, because no one neuron causes any action to occur. It's groups of neurons all signaling to each other and working together that cause us to be able to move, think, talk, breathe. All of these things are groups of neurons acting, and they lose the cells with the Lewy pathology lose many of their synapses. They lose functionality in their ubiquitin proteasome process, and that's just a fancy way of saying the cell's garbage collector. So all of the parts of the cell that are no longer being used and that are damaged or degraded, or they've been sticking around for too long, they all get sent to the ubiquitin through the ubiquitin signaling to the proteasome, where they're degraded, which it's just the dump of the cell. And what they actually lose is their ability to use their dump. And so all sorts of other toxic things build up inside of the cell because they're no longer able to get rid of it. And then lastly, what we see is loss of their mitochondria and metabolism function. So mitochondria is the powerhouse of the cell. It's how the cell produces energy. And what we see is that there's actually massive loss of many of the proteins involved in energy production. So the cells with aggregates are really, really sick. And actually what we see overall is activation of cell death pathways. These cells get very sick and they die. And so what I'm really interested in the next part of my thesis, because we were able to publish this work, it's now peer reviewed and it's published and publicly available, and all of my data is publicly available. So hopefully many researchers will be able to hone in on the gene targets we were able to find and find some new therapeutic avenues. But what I really am interested in the latter part of my thesis is understanding how exactly cells have this baseline vulnerability or resilience to pathology, and then trying to use all of the gene targets we found as markers of the pathology toxicity to be able to boost the resilience of cells kind of at a baseline. And so that's what I've been focused on since the early part of my thesis, which this work was focused on. And so let's recap. So not all neurons have Lewy pathology, Parkinson. And so what I've expanded on here is now we understand in the cortex specifically what neuronal subtypes are vulnerable and resilient to this pathology. And this allows us to ask those questions about, at a baseline, what factors are involved in resilience and vulnerability to this toxic cell death that occurs.
I guess I'm maybe not understanding this. You're saying that different types of neurons are vulnerable?
Yeah, exactly.
Okay, but I also thought you said that the ones that have the Lewy bodies
Mhm.
are genetically disrupted. So in other words, you're suggesting that there's a subclass of neurons that are genetically defective. Is that true?
So, well, there's all sorts of things that happen to the DNA at a baseline, right? So in Parkinson's disease, there are some genetic factors that contribute to the disease, and there's actually, we believe it to be a polygenic disease. So many genes may be involved in the kind of baseline predisposed risk to Parkinson's disease.
No,
What I'm talking about here specifically is that when an aggregate occurs, we see that actually causes damage to the DNA and to the RNA. And what this means is that even if the cell is able to kind of express proteins or express RNA, that RNA is all jumbled up. So
the body is causing the damage to the DNA.
The aggregate seems to be what's causing the damage. Yeah.
Okay, and
Jump in here.
the same thing is, sorry,
Well, I think part of your question too is it seems like some neurons are more likely to have an aggregate in the first place. So there's two things going on here. Some types of neurons, because of however their DNA is made up of, are a little bit more likely to get the aggregate. And then once they get the aggregate, they get further damaged on top of that.
so the ones that get the aggregate, you don't know what the trigger is.
Yeah, we don't know what causes that initial misfolding to occur. But what we've been able to show kind of, and this isn't me, this is kind of the field in general, is that the pathology seems to be able to propagate itself. And so even a small amount of an initiation event, so a very little amount of protein initially misfolding can then propagate through the brain.
Okay, and then
Yeah.
may I ask one last? As far as you can tell, the aggregates are driving mitochondrial to death and not opposite direction.
So
I guess I'm a mycologist, and so I'm familiar with killer strains of Ascomycete fungi.
Yeah.
Which have variable, mitochondria within the cell, and then eventually basically you get competition. So it's not the mitochondria causing the death, it's the opposite way.
it is the opposite way, and we were able to determine that kind of experimentally. So what we were able to do is in models of the disease, we induce the pathology, and then we see the mitochondrial dysfunction. So we're able to experimentally show that now, while mitochondrial dysfunction may exist in the other cells as well, the aggregate is specifically able to kind of cause this damage to the mitochondria. So the energy production gets really damaged by the formation of the aggregate.
Thank you.
Once those Lewy body neurons are damaged, is that, I mean, this is the question of the day, is that reversible? I mean, you got like 8 parts of that circle, so maybe the garbage part is damaged. Do we know what gets damaged first? Do we know, have we ever seen where we've had partial damage and some improvement, or do we see any differentiation between those Lewy body, like when you're looking for them and you're saying, okay, here's one that's got 5 damage in 5 areas. Here's one that only has damage in 2 or 1. Is a progression? Is it?
Yeah, so that's a fantastic question. It's actually a major part of how I've been following up on this, is understanding exactly that. Like, right, does this happen all at once, right? Does the aggregate form and now all of these things are damaged? Or is it
Function is
kind of
gone.
progressive over time?
Yeah.
And I can tell you it's not peer reviewed yet, and this is kind of preliminary from my work, but what we do see is that while the toxicity seems to be conserved, what happens, there does seem to be this trajectory of a progression over
Okay.
time. So we're still teasing that out, but it does seem like this is something that we can try and intervene in. right?
So a cascade?
Yeah, like a cascade, right? Like, so this all feeds into itself,
Okay.
this damage does.
Is it, for lack of a
better term, infective. And when you showed your scans of the brain,
Yeah.
the aggregates seemed to be clustered in a group of cells that were all, there were clumps of cells that all had the aggregates. Do you know anything about that?
Yeah, so I wouldn't call it infective, so it's not like, you know, you can catch these aggregates from someone else, that's not.
I'm talking about
But
spread
within the brain, yes, it does seem like one neuron can send the pathology to another neuron and then so on and so forth. So as I showed you in that schematic of the spread, that is along brain regions which are directly connected to each other by synapses, right? So the neurons all project to each other in those regions. So it seems, at least to our best understanding as it is today, and there's some variability in this, but it does seem that it is a progressive, almost infectious like spread where it goes from cell to cell.
Yeah, I mean, actually, that curve you show with the, you know, I do plant pathology.
Yeah.
And that's a typical disease epidemic spread. And that area that you highlighted is the time when you start to get epidemics. So, I mean, I'm just wondering if you could think of it that way, essentially.
Yeah,
A disease epidemic in your brain.
Yeah, so actually, one of my projects is looking at exactly that, where we use math models of spread to predict pathology, and then we look at how well that math model of pathology spread predicts what we actually observe in the pathology spread. And so we do use exactly that kind of mindset to understand how this pathology is kind of propagating itself. So yeah, it's a really cool element of how we're able to understand this. And then by understanding kind of how these models may predict spread, we can also target those key nodes to that spreading process and hopefully slow it down.
Yeah, can I just clarify, the neurons that you found to be vulnerable, were they the excitatory?
Yeah, so these are indeed excitatory neurons. So they're neurons that when they signal, they are likely to cause other neurons to be more excited, right? Signal more. Yeah.
And is there something significant in the fact that and so the resilient ones were the inhibitory neurons?
Yeah,
And
and I really thought that might be the case, but then what I found is that these PT neurons, the pyramidal tract neurons in layer 5, they're also an excitatory neuron type. And so it doesn't seem like it's specifically this excitatory inhibitory. It seems there's some sort of very specific factors that are contributing to, you know, resilience and vulnerability that I'm still trying to tease out in my project, and I'm really excited to kind of dig into that deeper in this last part of my thesis project. That's yeah. Thank you.
So if I understand what we just said from a layman's person, are we trying to figure out if this neuron can be replaced or fixed to make this stop or possibly go backwards?
That's exactly what I'm trying to do. I'm trying to see what parts of this can I fix, and will that help slow actually disease modify, right? So this isn't targeting ways that we can treat symptoms. This is targeting this underlying biology, and hopefully we'll be able to find some targets that allow us to actually modify disease, because this is the biological process that's happening instead of just the symptoms of the degeneration.
May I ask one last question?
Can I come to the microphone? Just as well.
It appears that
It appears that one of the crucial things is to understand what triggers aggregation. Do you know that yet?
So we don't know what triggers the aggregation. We know of ways that we can take alpha-synuclein, put it in a test tube, and make it misfold. But exactly how that happens in a living system, we don't understand fully. So we know ways to make it happen, kind of, we call it ex vivo. So outside of a living system, we can make it happen. But why that happens inside the living system, we don't know. And there's actually some people who are kind of hypothesizing that with this protein in particular, it seems like it's just predisposed to take on many different conformations, many different shapes. And one of those shapes just happens to be this misfolded propensity towards aggregation shape that then kicks off.
So you're not advocating for any environmental stimulus?
So there are definitely environmental toxins that can induce Parkinson's disease. We know that very well. And there's environmental toxins that can induce this pathology as well. But we don't think that that's a universal factor. There are certainly people who haven't had exposure to those toxins who do get Parkinson's disease. And so this is one of the ways we understand the disease. But what I'm trying to target in my work specifically is what things can we try and do to really target this process as it's developing and try and slow down the disease.
Okay, one last question, I promise.
So
Are all of the neurons producing these aggregates, but the ones that become pathological, they're the ones that are producing the aggregates at a much faster rate?
So we're not entirely sure on that. We think that all of the neurons seem to be exposed to these aggregates, right? So you have a neuron that has a ton of these aggregates. It's got this big Louis body, and it's kind of sending the misfolded alpha-synuclein to other neurons. And some neurons, when they're exposed to that misfolded alpha-synuclein, then develop those large aggregates themselves, and other neurons don't. But it's not entirely clear that every single neuron is equally exposed to the pathology.
One more question behind, Ashi. So this
This is
is
going to just be an open-ended, and I have no idea where to go with this, but is any thought given to autoimmune function in between the way the human's immune system is working and the difference between one neuron's response and another being some type of autoimmunity disorder?
Yeah, so I'm not an immunologist, so I'm gonna first off
Yeah.
open up with I'm not an immunologist, so I'm not really an expert in that area. But it does seem that the immune response is really critical to this process, and that's the kind of innate immune response of the body. And so actually one of the areas where I'm following up on right now with I'm hoping to do right now, I'm validating that this is a viable pathway, is actually an immune pathway where I'm trying to investigate that pathway and its involvement and whether or not we can really drug that pathway to try and alleviate the toxicity of the aggregate.
I think I heard you say that one of the questions was it trying to help or was it contributing to the problem? Is that the ace then? You're sort of asking that question about it.
Yeah.
And because I've seen that question sort of in the wider field. And how did you determine the answer to that question?
So what we found is like all of those different dysfunctions that I highlighted in that circle figure, that was all things happening in the aggregate bearing neurons, right? So the upregulation of DNA damage and the immune response, and then the loss of all these really key cell functions. So that tells us that it's toxic, right? And so it seems like at least at these end stages where you have these large aggregates, right, that it is a toxic function. Now, early on, it may be a protective response. So that's not entirely clear to the field yet. But it seems at this end stage where we have these large aggregates, it is toxic. And we got one last question.
Hi, this is definitely a layperson's question.
Awesome.
And the answer might be, it depends, but
Usually is in biology.
if a person's diagnosed with a Parkinsonian condition and not specifically Parkinson's, how much of this pathology might align?
So that really does depend. So right, there's these. So predicted my answer. Like someone had alluded to before, there are these environmental toxins that you can be exposed to. Some of those environmental toxins will actually induce this pathology, but others don't. And then there are also genetic forms of the disease where you don't observe this pathology at all. And yet you still get many of the exact same symptoms of a Parkinsonism. And so we call this Parkinson's disease. And so right now the field's talking about shifting the way we talk about Parkinson's disease to talking about these different forms where you may have pathology, you may not have pathology, or you may have like an environmental toxin. And so right now I'm talking about specifically Parkinsonisms that have the pathology. And this is what we've found in our research is that for the Parkinsonisms that do have Lewy pathology, the Lewy pathology seems to be inducing the same toxicity to the cells.
Fantastic, thank you, Thomas.